Welcome to the second season of Light the Clock. We are back and ready to illuminate you on all things light and circadian rhythms. And we're kicking off with a journey through time. If you were to open a classic chronobiology textbook,
Here. Chapter 1, History of Chronobiology.
you would probably expect to find the name of a human biologist. Someone, in other words, studying biological rhythms in humans. Well, you gotta think again, because you're likely to stumble upon the work of a French astronomer from the early 18th century.
Je suis Jean-Jacques Dortout de Méran.
Not peering at stars, but rather experimenting with plants. So, what was this astronomer up to, and what does that have to do with chronobiology? Well, to find out, I called up our expert, Professor Orie Schafer.
Dialing in now. Let's go.
Yes, of course. And first of all, thank you very much for the invitation. It's a pleasure to be here. So I run a research lab at the Advanced Science Research Center at the City University of New York. We're in West Harlem, New York. And my lab studies the neural basis of the circadian clock, so the clock that times your sleep-wake cycles, how environmental light resets that clock, and how that clock controls sleep.
Cool, thanks a lot. So today with you, we want to take a little step back and go back in time and delve a little bit into the history of chronobiology. So this is the field that studies biological rhythms. So we talked a little bit about chronobiology before in the podcast, but we didn't really talk about the first experiments or when it all started, actually. So when did researchers actually start to suspect that we had an internal biological clock? And in which organism was this? Was it in humans, in plants?
Yes. As someone once said, it all started in plants. So really, from the beginning of recorded history, you have very observant people seeing daily changes in plants, particularly in their leaf movements and in their opening and closing of their flowers. And so this had been appreciated for a very long time. And in fact, these were referred to as sleep movements of plants. They suggested that it had some relation to sleep. But the question is, when did we really start to do experiments on this? And this started in the mid-1700s, early to mid-1700s, in plants, in sensitive plants. So these are these beautiful plants, mimosas, that when you touch them, they quickly close up their leaves. And these plants show daily changes in those leaf movements. So they close at night and they open during the day. And it was really, remarkable early experimentation on that species that started the field of chronobiology.
Okay, cool. And so what kind of experiments were done on these mimosa plants?
Well, the most famous is by a natural scientist named de Moran. I'm probably mispronouncing that.
He's French, isn't he?
Yes, Myran, I think. And so he took these mimosa plants that were showing these daily opening and closing of their leaves. And the assumption was that this was just driven by increases and decreases in light as the day passed into night. And so what he did was he took his mimosa plant and he locked it away in a cupboard, away from environmental light. And he peeked in throughout the day and noticed that the opening and closing continued inside that cabinet, despite the fact that it had been shielded from the light-dark cycle of the environment.
Right. And so there must have been something else that was driving this, right?
The idea was that there must be something else. Now, de Moran did not hypothesize the existence of a clock. And it was really hard to believe until quite recently that such a thing would have to exist on a rotating planet. So I think initially the idea was, well, it wasn't driven by the light, but it could be driven by other environmental changes throughout the day. And it took about 100 years of people following up those experiments until de Candolle in 1832 published these results where he put the mimosa under constant light. And he showed that the leaves continued to open and close, and really, really amazingly discovered that the rhythm under constant light was faster than 24 hours. he was really, I think, the first to show that there's an endogenous rhythm that has a period that's slightly different than the 24-hour solar day.
Okay. And so now we're like mid-19th century, right?
Yeah, it took about 100 years between Desmarins and Descandolle to really see the first evidence that it's endogenous, that it's not just a 24-hour rhythm driven by the rotation of the planet.
Okay. And so now we know that this happens in plants. And when did scientists actually start to think, okay, maybe this also happens in other organisms as well?
Yes. So, it started in plants, but it went into insects pretty early and into other animals. But this persistent skepticism that there is such a thing as an endogenous rhythm really survived for a very long time, despite growing evidence from other plant species, simple animals like flies and cockroaches, and then the incorporation of rodents. But yeah, by I'd say the 1950s, 1960s, it was pretty clear to most people that the majority of animals, plants, and many fungal and even unicellular organisms have these rhythms.
Right. And so I guess by this time we have like accumulating evidence that these internal rhythms that are independent from the outside night and day cycle exist. I guess at this point, because humans are also animals, right? Scientists started to look into humans.
Yes. And so those initial experiments were really quite adventurous. So the initial experiments involved, descending into caves, far underneath the Earth's surface and asking questions about how sleep cycles function there and taking careful, measurements, usually the scientist measuring himself. And yeah, it became pretty clear that even deep underground, under constant temperature, no light, there were daily rhythms in sleep and activity in human beings. Now, this was followed up in an extraordinary way by the creation of the Index Bunker Laboratory, started by Jürgen Aschoff, where he, brought human subjects into a very tightly controlled laboratory situation where they were living underground with the ability to measure their daily rhythms and behavior and physiology with exquisite detail, and also to really carefully control the environment to make sure that those human subjects were in complete temporal isolation.
Right. So basically now it's kind of like the old experiment with the plant, but we're locking, quote unquote, humans into a cupboard, right?
Exactly. This is the human equivalent of the sensitive plant, of putting a sensitive plant in a cupboard in the basement. Yes.
Right. And so what was found out there? So I guess knowing what you just told us about plants and other organisms, we would expect that then humans in these constant conditions also have like this internal clock that runs around 24 hours.
Exactly. So, it shouldn't surprise anyone who understands evolution that all of the hard work that had been done in other mammalian species using things like running wheels or birds on perch hopping rhythms and things like that there was clearly an endogenous rhythm, that the daily sleep activity cycles and temperature cycles of core body temperature did not require any access to the time cues on the surface of the planet, that they continued to run very, very accurately, albeit with a period that was significantly different than 24 hours.
Right. And so these participants that were in this bunker, which by the way it's not far from where we're recording the episode, right? I guess I'll put a link in the show notes for listeners to go and check out the pictures, because there are still really good pictures of it. What were these participants doing in this time, and how long were they in the bunker? Was it like complete darkness?
It really depended on the conditions. I don't know if they did complete darkness. I'm not sure that would have been ethical,
because that's really tough, right?
Yes, the initial subjects were the investigators themselves. I know Dr. Aschoff was one of the first subjects of those experiments. But also, it's a proud tradition at universities to use your undergraduate population as experimental subjects. And I know that there were a lot of undergraduate students from the university there who volunteered. Evidently, they thought it would be an excellent opportunity to get a lot of studying done. And so these are very simply laid out apartments that had a place to sleep, a place to study. I think they had a record player, and so very simple but comfortable apartment. And I think some of the experiments lasted quite some time, well over a month, and sometimes I think maybe up to 100 days.
Wow, that's crazy. Now that we found out that also humans have this internal clock. Did researchers start to think a little bit about where is it, right? So it must be somewhere in our body. Is it in my nose? Is it in my ears? Is it in my legs? Yeah.
Oh, absolutely. And actually, this was actually a really, the relationship between the mammalian circadian system and light. So it was clear from all the experimental work on animals that in animals, you have this near 24-hour rhythm. It's precise but inaccurate, and it needs to be reset every day to stay on local solar time. In mammals, we had the advantage of the brain receiving light input from really only one source, and that's the retina. Other vertebrate animals have multiple ways for light to get into the brain. But in mammals, because of the nocturnal bottleneck, all existing mammals evolved from nocturnal mammals. For whatever reason, that was accompanied by the loss of everything but retinal input. And so by following light input in from the retina, chronobiologists had a pretty good idea of where the clock might be. And it was really experiments in rodents of basically ablating small islands of brain tissue and figuring out which ones were required for the clock that chronobiologists were able to locate that clock in the brain.
Yeah, which is, we've mentioned this before on the podcast. This is this little grain of rice, we called it, in the brain, right?
Yes, the suprachiasmatic nuclei. Yes, exactly.
Very cool. So once we found out where it was, I guess researchers also started to wonder a little bit about how it works, right? So because it regulates a lot of our physiological functioning, it must be somehow encoded in our biology, so kind of innate in our DNA.
Yes, I mean it's such a striking phenomenon. Really unexpected. I mean, there was still some pretty deep skepticism that one should need a clock on a rotating planet. But once it was established that there was an endogenous pacemaker in the body, it was clear that it must have evolved. It must have afforded some selective advantage and therefore must be heritable. And if it's heritable, it's probably determined by genes. And so the race was really on. Well, it wasn't really a race initially, right? The idea was that there must be genes involved and that one could use modern biology to find those genes.
Right. So I guess when we experiment on genes, which organism are we experimenting on?
There is an undeniable star to this story, and that is
your favorite star.
My favorite star, Drosophila melanogaster, commonly known as the fruit fly, more accurately known as the vinegar fly. This has been an undeniable powerhouse of modern biology. And that's due to the fact that it's a wonderful organism for discovering the genetic basis of almost any aspect of life that you're interested in.
Okay. And so how are these first experiments on the circadian genes, what were they like?
So now this is based on earlier work by a really important character named Colin Pittendrigg, and he was following up work by someone named Kalmus, who first used the fly to study circadian rhythms. And so there was a lot of foundational work on circadian rhythms, the formal properties of circadian rhythms that had already been conducted by Pittendrigg and others in Drosophila. This was a different species of Drosophila. But in the 1960s, 1970s, an amazing investigator named Seymour Benzer decided to use what's called a forward genetic screen. So this is really what makes flies amazing, is the ability to do what's called a forward genetic screen. Would you like me to lay out what that is?
That'd be great.
So this is really the key to why the fly was so informative. And so what a forward genetic screen is you take thousands of flies that are essentially genetically identical, and you feed them a chemical mutagen. So this is a chemical that when added to the food will tend to cause a single letter change in DNA. So one and only one change usually. And so what you do when you feed those flies that mutagen is you create a large pool of potential mutants. And some of those mutants will show changes in the process that you're interested in. And for many, many years, this was used to understand development. You would feed flies these chemicals and then look for flies that didn't develop properly. You could then map that gene, identify the gene product that it makes, and then learn something really important about how development functions, how the body plan is laid down.
Right. So you can basically understand from like mutating this single letter, you can pinpoint a gene that is responsible for development in this case.
Exactly. And so what Seymour Benzer decided to do was use forward genetic screens to understand the genetic basis of behavior. And so he created his lab around this idea. And his first graduate student was a guy named Ronald Konopka. And he, based on his understanding of Colin Pittendrig's earlier work, said, well, I'm going to feed these flies mutagens and I'm going to find fly mutants whose clock doesn't run properly. And what he did was spectacularly successful. He concentrated on one chromosome, the X chromosome, and he found 3 mutations that all mapped to the same gene that he called period 0, period short, and period long. And these were changes in a single gene that either stopped the clock, made the clock run really fast, or made the clock run really slow. And this was really the birth of clock genetics.
Right. And so basically what his finding is that this period 0, this gene that you mentioned, is responsible for the normal 24-hour internal clock function.
Exactly. So, well, it was really critical that he found not only a mutant that stopped the clock. So period 0, if you put flies under constant darkness and constant temperature, so this is the Andex bunker experiment for flies, you put them under constant darkness and constant temperature, they will continue to have a near 24-hour rhythm in their locomotor activity. Per 0 showed no indication of any rhythm under constant darkness and constant temperature. So once you remove the light-dark cycle, they no longer showed a daily rhythm. But it was really critical, actually, that he found the other 2 alleles, per short and per long, because these were flies that had a rhythm. They continued to maintain that daily rhythm, but they did so with an 18-hour period or a 27-hour period, which really showed that he had tapped into the core mechanism. There's all kinds of ways to stop a clock that isn't hurting the clock. The animal could just be really sick or confused or hyperactive. But by showing that he could mutate a gene and not abolish the clock, but actually determine its speed, was really the clear indication that he had found a core clock mechanism.
And what would happen if you put these flies that have a very short or a very long period? If you put them under a 24-hour light-dark cycle, what happens then?
It's a great question. So as it turns out, everything, if say you take the per short mutant, everything happens earlier. So flies are like mosquitoes. They're active in the morning and at night. Their activity anticipates dawn and dusk. And if you look at per short under a equinox, under 12 hours of light and 12 hours of dark, their morning and evening peaks happen earlier than they would in a wild-type fly. And the converse is true of per long. When you slow the clock down, the timing of daily behavior is delayed as well.
So going a little bit more in depth into Drosophila research, which is the focus of your research, I think listeners will be thinking, okay, these circadian rhythms, this time setting that we do every day in humans, this is all quite complicated. How can you model that in a fly? How can Drosophila be like a very good model organism for that? You mentioned before that you can study molecular bases because you can manipulate genes very easily, which in humans it's a bit harder. So, but for other things, rather than like understanding the gene function, how can you use the fly to model circadian rhythms?
Well, if I could tell you a story about a really remarkable story about the relationship between the fly clock and the human clock, you could always cut it out if you want to.
Please.
So I just mentioned the per short mutation that Ronald Konopka found. So this is a single nucleotide change in the DNA, a single amino acid substitution in the protein that causes the fly's free running period to go from, say, 23.5 hours to 17 and a half hours per cycle. So it's really, really sped up. And you just asked, what happens to that fly if you put it in the real world, if you put it under a light-dark cycle? And what happens is when it wakes up and when it falls asleep happens much earlier in the day. Well, fast forward, I think maybe mid to late 90s, in Northern Europe, human subjects were showing up to sleep centers saying they had a very strange problem, that they were falling asleep before dinner time. They had a very, very hard time staying up long enough to have dinner with their spouse. They were waking up extremely early, and it didn't really matter what they tried to do. They really couldn't reset their schedule. Long story short, those are per short human mutants. Those are single nucleotide changes in their version of the period gene that was discovered by Ron Konopka. And they suffer the same problem. Their clocks run too fast, and everything happens too early in the cycle. So this is a really clear indication that what we discover in flies can be amazingly relevant to what happens in human beings.
Oh, that's really cool. And so are there also individuals who have the other mutation that makes them have a longer period?
That's a great question. So Michael Young at Rockefeller has found mutations in other genes called cryptochromes, and those cause delayed sleep phase syndrome, and they slow the clock down and they make everything happen later. And as it turns out, a pretty significant proportion of humans have these variants.
Some of your work focuses on aspects of modern lifestyle that can influence circadian rhythms. So, for example, light exposure at different times.
Mm-hmm.
How can you study these things in Drosophila, so in a fruit fly? I guess you don't have them scrolling on a screen, on a smartphone.
No, we haven't trained them to use smartphones yet. Yes. So, and it's a challenge too. Well, in a way, it's interesting to study light input in an organism that can't close its eyes. So that makes it a little easier for us, actually. But it remains a powerhouse model organism simply because you can run thousands and thousands and thousands of animals through lots of different light-dark cycles and examine their sleep-wake cycles and their circadian rhythms. So just the sheer numbers of subjects that you can put through many different carefully planned light regimes makes it a wonderful model system. And then our ability to change the brain or change the eye or change the clock using modern genetic tools that allow us to make very specific changes in the brain, allow us to test hypotheses, to test models in a really direct way that's not feasible using vertebrate models.
Something that might also be interesting to the listeners is knowing what is the difference between Drosophila, so this fly that you have in the lab, to just flies that exist in normal life, so outside.
Sure. Well, chances are you've got this very fly in your fruit bowl at home at this very moment if you've got fruit sitting out. So it's very common. So it's cosmopolitan now. It's on pretty much all continents except down in Antarctica. So you have absolutely seen these. The reason why they're called fruit flies is because people encounter them very frequently in their ripe fruit at home.
Okay.
So this is certainly a fly that you've seen. It is not the large house fly. It's the tiny little fruit fly that you see buzzing around your bananas at home.
That's really cool. So I guess listeners can have a look in their fruit bowl and see this little fly.
Yes.
And kind of think that the first experiments investigating the biological clock were done on this very fly.
Certainly the first genetic experiments, without a doubt. Yes. Yeah. And I think this highlights another wonderful thing about the fly. They're extremely easy to raise. You're raising them right now at home, whether you want to or not.
Yes, that's great. I think we can wrap up. Orie, thank you so much for joining us on the podcast.
Thank you very much for the invitation. I enjoyed it very much.
Me too. Thanks to Orie for kicking off Season 2 of Light the Clock and bringing us back in time to the early experiments of chronobiology. And thanks to you listeners for tuning in to today's episode. If you're curious to see how the bunker experiments in Germany looked like, you can head to our show notes. You'll also see other interesting resources there, including a link to Orie's webpage, so a link to his research group. If you have any feedback or even ideas for future episodes, please reach out. We would be super happy to hear from you. You can either find our email in the show notes, or if you are listening on Spotify, you can also ask us questions directly in the Q&A box, which is a fancy new feature which we activated. By the way, you can now also follow us on Instagram. Our handle is Light O'Clock Podcast. That's Light O'Clock Podcast. All lowercase, no symbols, nothing, just plain. On Instagram, we post more info on our guests, as well as summaries of the episodes. So if you feel like you're missing some visual content when you're listening to us, like how does the light spectrum actually look like? I need to see this, not just listen to this. Well, my colleague Zara has created some fantastic visuals that summarize our episodes. So you can head over to Light O'Clock Podcast on Instagram and check them out. The next episode will be coming out in 2 weeks' time, as always. And we will be chatting to Dr. Renske Locke about the relationship between circadian rhythms and the sleep-wake cycle. Are they the same? Are they different? Tune in to find out. You have been listening to Light the Clock. This episode was produced by me, Caro Guidolin, with help from Lucien Bickerstaff, who lent his perfect French accent to play Jean-Jacques Dortu de Meren. I'm not sure that's right, but we're gonna roll with that. Manuel Spitchen was our fact-checker. Music production, mix, and sound design were done by Ingo Bednare. Show notes and additional resources were created by Maidel Fernandez Alonso. Our social media team includes Hanna Sophie Heinrichs, Nilufar Tabande, and Sarah Alkaf. Lucien Bickerstaff helps with the podcast distribution, and Anna Magdalena Billa is our test listener. Light O'Clock is a podcast produced by the Translational Sensory and Circadian Neuroscience Unit, and we are supported by the Circadian Mental Health Network, the Viscom Prize from the Deutsche Gesellschaft für Psychologie and the Deutsche Gesellschaft für Psychophysiologie und ihre Anwendung, the Max Planck Society and the Max Planck Institute for Biological Cybernetics, and the Technical University of Munich.